Welcome back to BioMindscape. Here we are going to know about the comparative genome view. So here I just selected a species here like Homo sapien, human, and select an assembly here, and after that I have selected the second species, for example gorilla, and then I have selected the view the comparison. I am just clicking on the view comparison. Let's compare. So the Comparative Genome View is an interactive web-based visualization tool used to compare a whole genome assembly alignment of two eukaryotic organisms. It highlights the structural similarity, chromosome rearrangement, and conserve the gene order across the species. So ideogram display shows the chromosome side by side or stacked using a colored ribbon to match the matching alignment segments and orientation. There is a Synteny tracking that helps the researcher visualize the conservation of the gene block and order the difference between the genomes. Here, the structural variant detection, it quickly identifies the large-scale genomic events like inversion, insertion, deletion, and translocation. And there's a data export that allows the user to download a specific alignment segment in the FASTA format or generate a vector graphic (SVG) images. So this is a whole genome alignment visualization from the NCBI Comparative Genome View. So it maps the chromosomal region between the human and gorilla. So first the top axis it represents the human genome assembly, and the bottom axis it resemble the gorilla genome assembly, and the connecting line it shows the region of the high sequence similarities alignment between the two species. So the green line it represent the forward alignment, purple represent the reverse alignment. So here the genetic sequence matches in same direction on the both and human and gorilla in the green alignment, forward alignment, whereas in purple alignment we can see the genetic sequence matches but in an inverted flip backward in one species compared to another. So in the human chromosome 2 fusion, you can see the human chromosome to connect to two distinct chromosome in gorilla labeled 2A and 2B in standard karyotypes through number sequentially. So this is a direct visual evidence of the evolutionary events when the two ancestor ape chromosome fuses to form the human chromosome 2. The large inversion, the chromosome with a heavy crisscross purple line indicate the large scale chromosomal inversion. This means the large block of the DNA become flipped during the evolutionary split between the human and gorilla. There's a dot plot view. So in the dot plot view is an alternative 2D matrix layout used to visualize the large scale genome alignment and structural variation. Clicking to the go to the dot plot view, we can see the x-axis axis one genome assemble example human is plot along the horizontal x-axis and other genome y that is vertical plotted for the gorilla. So here there is a diagonal indicator, forward direction, anti-diagonal indicator, inverted alignment, and offset line on breaks. It shows the insertion, deletion, or translocation where the section of DNA have shifted the position. So anti-diagonals it show chromosomal inversion. Diagonals it is indicating the forward alignment where the sequence share identical orientation across both the species. For finding a gene in a comparative genome alignment, we have to type the name of the gene which we wanted, like a LOXHD1, that is a human ear gene comparing the gorilla and human ear. So let's see the alignment. Let's go to the search bar and search it. So we get the assembly gene LOXHD1, their location as well, like chromosome 18 and chromosome 17. So we can know that particular genome as well. Here LOXD1, where are they present in this particular view? Here we can see LOXHD1 at various sites. This is for chromosome 18. And at the downside we can see for the chromosome 17 both are aligning with each other. The locks HD1. So your alignment details are given here, and we can zoom in and zoom out. Thanks for watching. Do like, share and subscribe BioMindscape.